Hello folks. We're going to spend some time studying cells in this course. So this presentation introduces the themes and some techniques for studying cells. The take-home message is that multiple techniques and approaches are used in concert to help us understand cell structure and function. From these illustrations, which you should have seen before or something very much like them, cells come in different sizes and degrees of organization. Eukaryotic cells are typically about 100 times larger than bacterial cells, and they show much more internal structure and organization. We call that a division of labor. Many of the structures in cells are organelles, which are membrane-bound compartments that segregate specific cell functions and chemical reactions. Much of the semester will be spent learning what these parts of the cells do and how they do it. The three main techniques we have for studying cells, generally speaking, are microscopy, which allows us to see the cells and in some cases to see where certain cell functions occur. Cell fractionation, a kind of microdissection to separate cell structures and compartments. And then, finally, molecular and biochemical analyses that identify molecules and reactions carried out by different parts of cells. Let's get an overview of several kinds of microscopy. In effect, different ways of seeing cells. The oldest way of magnifying cells is light microscopy, which includes traditional bright field, as well as phase and interference contrast microscopy, and fluorescence and confocal microscopy. Magnifications range up to about 1200-fold, 1200x, with a resolution of 0.2 nanometers. Resolution is the ability to discriminate between objects in the microscope. A resolution of 0.2 nanometers means that you can distinguish between two small objects that are, in fact, 0.2 nanometers apart. Objects closer than that will blur into a single fuzzy object in the microscope. So let's focus now or concentrate on bright field microscopy, which is typically applied to non-living, that is to say fixed, sectioned or sliced, and stained tissues. There are exceptions, however, but mostly it's fixed and stained tissues that have been sliced for microscopy. The stains provide color contrast, allowing the identification of cells and cell parts. The example on the left, illustrated here, or actually it's a photomicrograph, is of a plant cell showing the organization of the cells into different tissues. The high magnification micrograph on the right shows fully condensed chromosomes about to be separated during mitosis. Let's take a look at phase contrast or interference and interference microscopy. Across the bottom are three examples of living cells looked at in microscopes. On the left are intact but unstained living cells looked at in bright field microscopy. There isn't much to see because there's not much contrast between adjacent cells or between structures inside the cell. The cells can clearly be seen in the phase contrast micrograph in the middle panel, and each cell even shows a nucleus and hints of grainier cell components. The higher magnification of cells seen with interference microscopy on the right is even crisper and sharper, and reveals not only the central nucleus in each cell, or in these two cells, but other structures, the larger of which are probably mitochondria. These kinds of microscopy can be applied to fixed but unstained cells and tissue sections. But it's our ability to look at living cells by phase contrast or interference microscopy through which we can see how cells behave, how they move, and how they respond to stimuli. That is the true strength of phase contrast and interference microscopy. Now let's look at fluorescence microscopy. In this kind of microscopy, specimens on a slide are treated with a probe attached to a fluorescent dye. The probe can be a chemical that recognizes a specific molecule or structure, either in a cell or on a cell, and therefore binds to it. Typically, ultraviolet light will be beamed through the specimen, which is sitting on the stage of a fluorescence microscope. The fluorescent probe can be detected as a color emitted by excited electrons in the specimen. And that color could be any one of several shown. In this case, in this example, we see green and red and sort of violet or bluish fluorescence. Wherever you see the color, that's where the molecule or structure is that you're searching for. In this example, the specimens on the slide on the microscope were actually exposed to 3 different probes, each attached to a dye that fluoresced a different color when excited by UV light. The blue fluorescence comes from a probe that detects chromatin proteins, that is, proteins attached to DNA in chromosomes. The green and red dyes are really come from 2 probes that are detecting 2 different genes, 2 actual genes in the DNA. Hence you see all the colors are on the chromosomes of the cell in the middle that's actually in the process of mitosis and whose chromosomes have condensed. You also see the color through the nuclear membrane in the nuclei of the 2 cells on left and right as part of the chromatin in the nucleus. Confocal microscopy is a relatively recent invention of the last, I forget, 10 or 15 years. It's a special application of fluorescence microscopy that allows us to look at very thick cross-sections of tissues and cells, or even whole organisms, especially if they're small, like the embryo of a fruit fly. The microscope is focused at different levels of this thick section or organism, and an image is registered and then transmitted to a computer. The result is a set of computer-generated serial cross-sections that allows us to build a 3-dimensional image of the cells, or of the tissues, or even of organisms, that includes the location of specific molecules. In the image on the left, you can see some fibers of the cytoskeleton fluorescing green and lying over the nuclei, in which the DNA is fluorescing blue. Once again, because this is a form of fluorescence microscopy, it is possible to localize more than 1 probe at a time. In the mitotic cells at the right, once again, 3 different subcellular components are being localized, as you can see, because of the 3 different colors, the red, the green, and the blue colors. You might be able to look and figure out what is actually being localized. Blue is localizing either DNA or the chromatin on chromosomes at the metaphase plate of these cells. The red or orange fluorescence is probably coming from probes that are identifying some component of centrioles, which are the focal point of the formation of spindle fibers. And the green is actually recognizing the spindle fibers themselves. It's recognizing microtubules. There's a link at the bottom of this slide. If you want to copy it down, you can type it into the URL of your web browser and see and learn a bit more about confocal microscopy than is possible to tell you about here. There are 2 kinds of electron microscopy, or EM, tools for enlarging images of cells beyond the capacity of light microscopy. In transmission electron microscopy, we can get up to 1,000,000-fold magnification, 1,000,000 times, 10 to the 6 times magnification, and a resolution down to 2 nanometers. That's 1,000 times better than light microscopy. Scanning electron microscopy, shown on the right, can magnify in the range of about 100,000 down to a resolution of about 3 nanometers, so slightly less than transmission electron microscopy. In the transmission electron microscopy, or TEM image on the left of a bacterium, what has happened is a beam of electrons has passed through a fixed and therefore non-living specimen that has been treated or stained with an electron-dense dye, meaning a dye through which electrons do not pass. The parts of the cell that have picked up the dye appear dark in the electron microscope as a consequence. This TEM of a bacterial cell shows the paucity of internal cell structures. But do note the fine hairs or pili that coat the surface of the cell. These are helpful in the ability of the bacterium to recognize certain environmental chemicals or other cells. In the scanning electron microscope image on the right, we are looking at pollen grains, different pollen grains from different species. What happens here is the samples are fixed and then sprayed to coat the tissues with a very fine layer of rare metal. The SEM, the scanning electron microscope, beams electrons at the specimen, which are then reflected off the specimen, and the reflected image is captured by a CRT, basically a television screen from which one can generate these pictures. As you can see from this SEM, it's a beautiful tool for looking at the surfaces of cells and tissues, generally at lower power than transmission electron microscopy. It is even possible to look at whole organisms, at the surface features of whole organisms. You can Google scanning electron microscopy and see lots of examples of SEM images from very small things to very large things. To summarize, bright field and phase or interference contrast microscopy let us see the structures of and in cells. Fluorescence microscopy lets us associate specific molecules with structures, and confocal microscopy, a form of fluorescence microscopy, lets us see structures and locations of specific molecules in the context of a three-dimensional view of the entire cell or tissue or organism. Finally, electron microscopy is a really high-power, high-magnification instrument that lets us see the really small cell parts, and in some cases even some very large molecules. You will see that we can even use electron microscopy under the appropriate conditions to localize specific molecules to those very small cell compartments or cell components. Microscopy lets us see cells and cell parts and even the location of molecules, but we have to dissect the cells if we want to ask what specific parts of the cell do. This scheme for fractionating cells by differential centrifugation may look familiar. Cells or tissues are homogenized or ground up in a buffered solution intended to resemble the physical-chemical characteristics of cytoplasm. The homogenate is then centrifuged at low speed, or centrifugal force, or G-force, to sediment, we can also say pellet, the largest structures in the cell, in this case the nuclei. The liquid above the nuclear pellet is again centrifuged at about, say, 15,000 times G. These numbers are approximate and vary depending on the actual cell fractionation, but perhaps the next level of cell fractionation would occur at 15,000 times gravity, and the pellet, the 15,000 G pellet, contains the next largest subcellular structures. In animal cells, this would be mitochondria. In plant cells, this would also perhaps include chloroplasts as well as mitochondria. You can follow the rest of the eukaryotic cell dissection to the last supernatant on your own, watching this slide. The cytosol, then, is the last supernatant, and it contains all of the soluble components or chemicals in the cytoplasm, and no longer contains any of the particulates, any of the organelles or small structures in the cell. We'll have occasion to look at this kind of fractionation or the results of this fractionation again. Again, this is a cell fractionation or a cellular dissection. Take a look at this electron micrograph of E. coli. I'd like you to anticipate our next meeting by drawing or outlining a cell fractionation protocol for these kinds of cells in which you've done differential centrifugation to get the different cell parts. Give it some thought and have one ready for our meeting the next time. And here are the parts, the basic parts: plasma membrane, cell wall, the cytoplasm, the interior of the cell containing the cytoplasm, of course, which has mostly ribosomes, and the lighter region of the cell, which is called the nucleide, which actually contains the bacterial DNA, which is not associated with a lot of proteins. It's basically a circular double helix, which you may remember from another course. So again, devise a cell fractionation protocol for E. coli. Once we've separated the parts of a cell, we can ask, what do each of the parts do biochemically? The biochemical and molecular techniques we've developed over the last 75 to 100 years allow us to identify the particular chemical reactions that accomplish different cell functions. They allow us to associate specific reactions with specific dissected cell compartments. And they allow us to design probes to visualize molecules and chemical reactions inside the cells and tissues using some of the microscopic techniques I've just described. So this brings us to the end of this summary of techniques that we use to study cells. In this course, we're going to focus on how the different combinations of these approaches to studying cells has led to our current understanding of cell biology. And that brings us to the end of this presentation.